Hello everyone and welcome to the YouTube channel of the Biochem Research Group. In today's video I'm going to talk about a recently published paper concerning cosolvent and dynamic effects in docking calculations performed to identify new binding regions of proteins. To start off, some of you probably already know that computational methods are becoming increasingly popular in the field of drug discovery. With the exponentially increasing computational power that is available to us, these methods are now able to provide sufficiently accurate and quick results that are useful in a wide range of areas. In particular, computational docking calculations are utilized to predict potential binders for a number of biologically relevant proteins. The importance of these methods became even clearer with the COVID-19 pandemic, when they were used to study a number of important proteins for this virus and identify key inhibitors for them. Although undoubtedly they are very useful, recently one of their main weaknesses has been identified to be that they only consider a single rigid protein structure, which obviously cannot describe any sort of protein dynamics or flexibility. And this is a serious problem because these phenomena have been shown to be crucial in a number of ligand uptake processes, and protein flexibility also plays a crucial role in newer models of ligand binding, such as the induced fit or conformational selection model. It is also clear that by considering an ensemble of protein structures in place of a single one could improve the description of protein flexibility. However, with this approach, a new problem arises, and that is that it requires significantly more protein input structures. And obtaining such structures is notoriously difficult with the currently available expensive and tedious experimental processes. So it has been suggested that computational methods could be used to remedy this problem as well. In particular, molecular dynamics, MD, seems especially promising. After all, it has been designed with this very purpose in mind, that it should be able to effectively sample the conformational space of proteins. However, MD, at least traditional MD, comes with a sampling problem, which is illustrated on this slide, where the potential energy surface of a protein is sketched. The root of the problem is that traditional MD can only rely on small thermal fluctuations to induce meaningful conformational changes in the protein. Therefore, it can only sample conformations that lie low in energy, such as those on the left side here, denoted by these orange dots, one of which actually harbors an open binding site near the central regions of the protein. On the other hand, if one wishes to sample conformations on the right that lie somewhat higher in energy and are also behind a large energy barrier, then one has to modify the traditional MD scheme. Indeed, sampling these conformations could be very beneficial because, for example, here you can see that the leftmost conformation also harbors an open binding site that is completely different than the one already discovered. So to sample these conformations, one of the possible techniques is called Cosolvent MD, which I will explain on this slide. The main idea behind this technique is to replace the traditional water solvent with a mixture of water and some apolar cosolvent. This is depicted here on this top figure with the red cosolvent probes surrounding the protein. The idea behind this is that these cosolvent probes are expected to interact with the protein in a similar manner that a true binder would, and in this way they can help inducing conformational changes that would normally only occur in the presence of a true binder. So our goal during this project was to investigate the applicability of Cosolvent MD to generating these ensembles of protein structures that I've already talked about. Furthermore, given the severity of the COVID-19 pandemic, one of the vital proteins of this virus has been chosen as our prototype protein target in the hopes of contributing to the development of inhibitors for this protein. So from this point on, I will detail the results of the project, highlighting those that are connected to the applicability of Cosolvent MD. So first, the actual MD simulations had to be carried out. We decided to run 3 types of simulations, one with traditional water solvent and 2 cosolvent simulations with phenol and benzene as the cosolvent. With this choice, we have a nice gradient from most hydrophilic two most hydrophobic cosolvents. On the left side here, one can see equilibration root mean square deviation plots for the three types of trajectories. The equilibration of protein structures depicted in these plots is a crucial point in any MD trajectory, as it signals that the simulation samples relevant protein conformations without any artifacts such as the unfolding of the protein. It is therefore reassuring to see that the cosolvent simulations equilibrate just as fast or even faster than the water simulation. It is worth mentioning that we did encounter one problem with the benzene simulation, which was the clustering of the benzene probes, and this is undesirable because it reduces the interactions between the protein and the benzene molecules. However, this was straightforwardly solved by applying an artificial repulsive potential between the benzene molecules. After this, we were able to obtain the MD trajectories, from which we selected 19 representative protein conformations. And using this as our protein ensemble, we performed docking calculations to all of them with a library of about 2,000 small molecule drugs. The first statistical results of these docking calculations are shown here. Here, the binding energy distributions are separated according to the MD trajectory from which the protein conformation was obtained. It can be seen that the conformations coming from the cosolvent trajectories offer significantly lower binding energies, with the red and the green distributions reaching much lower. This also leads to the fact that a higher number of ligands with a favorable binding energy are discovered, as can be confirmed by comparing the numbers to the left of each distribution. So this first statistical result already indicates that the cosolvent trajectories could harbor more accessible binding sites, and it is reasonable to assume that these binding sites appear because of interactions with the cosolvent probes themselves. After observing that these conformations from the cosolvent trajectories are beneficial for docking, we also investigated whether using multiple protein structures is really worth the extra computational costs. To this end, the number of unique ligands discovered by the different protein conformations are visualized. The idea is that if all protein conformations just bind the same ligands, then it is not really worth it to perform dockings to all of them. On the other hand, if different conformations exhibit different binders, then the extra computational costs are actually worth it because we discover new ligands. So on the left side, the individual conformations of the various MD trajectories are ordered according to the number of unique ligands that they bind. This cumulative ligand binding plot visualizes the number of protein conformations that are necessary to identify a given ratio of the ligands discovered by all representative protein conformations of a given trajectory. It can be seen that considering a single conformation would discover about 50% of the binders for the water trajectory and about 75% of them for the cosolvent ones. Moreover, only very few protein conformations could be disregarded without sacrificing a significant portion of the identified ligands themselves. And then on the right side, a similar plot is shown, but this time all conformations are combined and treated. All conformations from all trajectories are combined and treated as a single unit. It can be seen that all types of MD trajectories discover new unique ligands, and therefore none of them could be regarded as redundant. This last data point corresponds to the crystal structure, and it can be seen that it discovers no new unique ligands that are not discovered by other MD trajectories already. So as a conclusion, one can say that the high number of protein conformations are not redundant because they discover new binders, and therefore their extra computational costs are well worth it. So after this analysis, we have also visualized the best binding poses for each of the binders. It can be seen from this figure that our calculations identified 9 major binding sites, which together correspond to 98% of all the best binders, which clearly highlights their importance for potential future studies. The active site of the protein is denoted with the number 4 here, and it is reassuring to see that a number of binders are found for this site, because this has already been identified as a druggable site. So after the pockets have been identified, we investigated the number of binders binding to each of them. So here you can see these numbers of binders separated according to the MD trajectory from which the protein conformation was obtained from. After investigating these numbers, we have identified two types of pockets, stable ones and transient ones. Stable pockets, such as the first two, bind a larger number of ligands, and they bind ligands in all three types of MD trajectories. So this signifies their stability, so the stability of their open conformation. On the other hand, transient binding sites, such as the third one, typically have fewer binders, and they are only open in perhaps one or two MD trajectories. In some cases, such as for number 3 and number 7, these open conformations are only found among the Cosolvent trajectories, which again highlights that these trajectories are able to provide highly druggable conformations. So to gain a deeper understanding of the opening and ligand uptake processes of these sites, a stable and a transient pocket, number 4 and number 7, have been more thoroughly investigated. First, the structure of the protein around pocket 7 is visualized. I remind you at this point again that this pocket 7 is only able to bind ligands when the benzene when conformations coming from the benzene trajectory are considered. So here the structure is visualized around this pocket for a conformation coming from the water solvent and one coming from the benzene cosolvent simulation. It can be seen that by shifting of protein residue 145 to the left and 155 downwards, a much larger cavity is created in the benzene cosolvent structure. And this shifting of the residues could very well explain why the pocket is only open in these benzene conformations, and it is also not unreasonable to assume that this shift occurs because of interactions with the benzene cosolvent probes. So as a comparison, we have calculated some pocket description metrics for pocket number 4, so for the stable pocket as well. On the top, you can see pocket volumes calculated for all conformations. It can be seen that a non-zero volume is observed for all conformations, confirming or verifying that this pocket is indeed stable. The pocket volume, however, still fluctuates significantly from 600 to 1800 cubic angstroms. Moreover, we can see that the red and the green cosolvent conformations are able to provide consistently higher pocket volumes, indicating that this pocket is more accessible in these cosolvent conformations. To further evidence this, we have calculated the hydrophobicity score of this pocket for the different trajectories. This hydrophobicity score aims to describe or quantify the druggability of binding sites based on the hydrophobicity and orientation of the surrounding protein residues. It can be seen here that this score is almost twice as high for the benzene and phenol conformations as it is for the water. So this indicates that this pocket is more druggable for these cosolvent conformations. So as a conclusion, this is ample evidence that using cosolvent simulations is advantageous even if well-known stable pockets are investigated, since these simulations are able to provide more open, more druggable binding sites. To conclude the talk, I will say that during this project, the advantages of utilizing cosolvent MD for the generation of protein structures ensembles has been shown. These simulations are straightforward to execute and have the same computational cost as traditional MD calculations. With the ensemble of protein conformations generated in this way, new binders of the target protein have been found, with many members of the ensemble discovering new unique ligands. And finally, Cosolvent simulations facilitated the opening of some transient binding sites of the protein and helped identifying new binding sites. Moreover, even if previously known pockets were considered, the conformations obtained from Cosolvent simulations proved to be much more druggable. So, regarding the future of the project, we believe that it would be beneficial to investigate a small subset of the discovered sites more thoroughly. In particular, we suggest that protein ligand enhanced sampling MD could discover even more information about the opening and ligand uptake processes of these pockets. So with this, I would like to thank you for your attention and see you next time.